Hello my friends, welcome back to my channel. In today's video I have very interesting for PhD and master students who are looking to publish a high quality paper in an easy and efficient way. This lecture focuses on the topic of from genes to functions, exploring genome-wide gene family identification using protein conserved domains. It's a fascinating subject that delves into the identification of gene families across entire genomes through the analysis of conserved domain. So without further ado, let's dive right into it. Here we go. In this lecture, I will be continuing my previous topic on finding intriguing genes for our research. This is especially important for students working in molecular biology who are seeking to increase their publications in an easily and efficient manner. I understand that many students face difficulties in certain countries where access to publishing in reputable journals can be challenging. That's why this topic is particularly valuable for them. In this lecture, we will delve into the concept of gene families and explore the significance of gene family identification. But what exactly is a gene family? Does a gene really have a family? Understanding gene families and their identification is crucial in molecular biology research. By studying gene family, we can gain insights into the evolutionary and relationship and functional similarity among related genes. Now, why is it essential to study gene families? Well, identifying gene families allow us to uncover patterns and similarity in gene function, which can guide our research and provide a foundation for further investigations. Moreover, publishing research in high-impact journals such as those with a Q ranking opens up enormous opportunity for recognition and collaboration. Throughout this video, we will understand and we will present several examples of publishing studies that demonstrate the significance of gene family identification. Additionally, we will explore the exciting possibility of discovering new proteins with potential functions through genome-wide identification. To begin, I will briefly introduce the genome-wide identification, then I will guide you through the step-by-step procedures for retrieving gene family on genome-wide scale. Get ready for an informative and enlightening session. Let's dive right in and explore the fascinating world of gene families and their identification. Here we go. The function of a cell and overall functioning of an organism are closely tied to the genetic information contained within the nucleus of eukaryotic cells. The genetic materials known as the genome reside within the nucleus and consists of billions of nucleotides. It's important to note that a significant portion of the genes present in the genome have the capacity to be translated into proteins. Proteins are the building blocks of cellular function, interact with one another, forming intricate molecular complexes. Through these interactions, proteins participate in complex metabolic processes which play a vital role in determining the identity and characteristics of the cell. It's important to understand that the genome composed of thousands of genes with distinct and unique functions. So, the gene can be transcripted to RNA and RNA can be translated to protein and this protein leads to a phenotype. So, it's very important to understand that the DNA and the protein sequence are correlated. The previous information drives us to the concept of protein conserved domain. We can retrieve the members of gene family in the genome scale using protein conserved domain. So let's understand the protein conserved domain and how we can use it to identify the gene family. Initially, I will explain what is the protein domain. The polypeptide sequence of a protein can be classified into different units. This unit, we call it the protein domain. This unit has unique structure and has unique function. So the function of a protein can be determined by a combination of these domains. We should know that during development and evolution, some domains with important function can be maintained, but can be recombined with other domains leading to produce new proteins. So we call this domain is a conserved domain. Accordingly, in one organism, we can find several proteins that share one conserved domain with potential function. So these proteins, we can say that they are belong to the same gene family. However, these proteins, they have distinct function. Additionally, the evolution studies have investigated that the conserved domain can be conserved in different organisms, leading to produce proteins with common function. However, this conserved domain make a good approach to find an interesting proteins in different organisms with potential function. However, but still we can find interesting protein with interesting function as they have a recombination with different domains. Several studies show that the protein sequences that are important for living organisms are conserved during the development. For example, the transcriptional evolutionary proteins are conserved in along organisms. So now I will show you how we can use the protein conserved domain from different organisms like multicellular eukaryotic to identify a gene family of histone acetyl transferase and histone deacetylase families from Arabidopsis, and also I will show you how we can use a conserved domain from Arabidopsis for genome-wide identification of a gene family from citrus. So at the beginning I will show you this paper. Actually it was published in Nucleic Acid Research. So I select this paper because they used known histone deacetylase and histone acetyltransferase protein sequence from varieties of eukaryotic organisms, as shown here in this table. For the genome-wide identification, they used the conserved domain from different organisms like Arabidopsis and maize, yeast, human being, and some Drosophila and some other animals. So now I will select a gene like a gene or protein from a human being to find some proteins that have the same conserved domain in Arabidopsis. So firstly we copy the gene ID from as shown here in the paper, and we go to NCBI, and then we plus in the NCBI, and we can see we found this gene in NCBI database. The annotation is histone deacetylase 1. So, and as we are working in a protein sequence, we can use a FASTA to obtain the sequence of this protein. So now we obtain a FASTA format of this protein, and we copy and go to the TER database, and we open TER database so we can make blast. As we have a protein sequence, so we'll select blast P. So blast P, it means that we use amino acid query for retrieving amino acid sequence, and also we have another option by blast N. By blast N we use nucleotide sequence as a query to find the nucleotide sequence as a hit. And also there is another option like blast X and the blast T blast N and T blast X. Now I will select blast P because I have a protein sequence as a query and I want a protein sequence as a hit. So we can run a blast, so we can waste some time to show the hits that has the same domain similar to that I used from a human being. So the result is showing that here are some hits showing the red color showing the high similarity, pink color is showing less similarity, and the green is more or less. So if we go to the annotation, you can see here from one protein sequence I found several proteins that have the same conserved domain, and this domain, if we see the annotation, is belong to histone deacetylase gene family. So now by this way I found all the proteins in the Arabidopsis that have the same conserved domain, and they are belong to the same family like histone deacetylase family. If I want to confirm and the protein sequence I use and what is a conserved domain, there is another tool. It's a smart tool. You can see here smart database. So this smart database, we can know what is a conserved domain in every protein. So as I have a protein sequence, so I select this option. So we need actually for smart domain we need a protein sequence in a faster format, so we can click these options, so we can search for we can search for different motif in different database. So we can select sequence smart, and we can waste some time to show the result and the conserved domain in our protein sequence. Here the result is showing that the protein I select from human being has histone deacetylase domain, and also here, so this is a conserved domain in different proteins in Arabidopsis. I want to show that this protein sequence that doesn't match the query, this is a unique motifs that recombined with the conserved domain. So this means that every protein maybe they have distinct function. This is actually what they mention here in this paper. So how they mention in this paper that when they make some bioinformatics analysis and compare between the protein sequence from Arabidopsis and animals, they found that even for this gene family, even the proteins from animal and Arabidopsis, they share a common conserved domain, but actually they have different recombination of motifs and domain. So this is making this protein has different function, and this is also was confirmed by the phylogenetic tree. So this is so that's why the title of this paper is Analysis of Histone Acetyltransferase and Histone Deacetylase Families from Arabidopsis thaliana suggests functional diversification of chromatin modification among multicellular eukaryotics. So by this paper they found even the histone acetyltransferase and deacetylase, they have a common and conserved domain, but actually they still every organism has some distinct function based on the recombination of the domains. I have another example about how we can use a conserved domain from like Arabidopsis to retrieve as shown here some protein some protein sequences. So I will select one protein sequence from Arabidopsis to retrieve the gene family from citrus Arabidopsis to run blast. Firstly, so I use the protein sequence of Arabidopsis to run blast in citrus pangenome database. So as I did in TER database, I will select here blast. So I will select, because I have a protein sequence as a query, so I will to find the protein hits, so I will select blast protein. I will select this database citrus sinensis, and then I will run and blast the protein sequence in faster format here, and I will run blast. Actually, most of database have the same way to find proteins. As we can see here, from one query of protein from Arabidopsis, I found several proteins in citrus that have the same conserved domain, and we can see this domain is very conserved. I'm showing here in the blue color, but still this protein have some distinct function in the N terminal. They don't share the same sequence. So by this way I explained to you how we can use a protein sequence and conserved domain to retrieve a gene family from another organism. So now I will show you the paper here by using this way. Actually, this paper I published in BMT Plant Biology was in Q1, is a Q1 journal, and with impact factor about 5. So firstly we can do some molecular properties of this gene family. What is the molecular weight? What is the subcellular localization? I will explain in details in next videos, so please follow me. And also I will explain how we can do the phylogenetic analysis, and then some phylogenetic relationship and the gene expression and the motif distribution of the gene family. And also we will explain how we can do chromosome how we can do the gene distribution in the chromosome. And also I will teach you how we can do heat map for the gene expression, and also here some data using heat map figures. And we can see easily this is the data I provide to be able to publish in BMT Plant Biology. It's just depend on some based on the identification using some bioinformatic tools, and just by gene expression analysis. I hope this video to be useful for you. And please subscribe and share this video with your friend to get some benefit, and write in the comment if you have any question. Thank you so much.